Welcome to this video assignment on DNA mutations. In the assignment, you'll determine whether or not a particular DNA mutation will have an effect on the resulting amino acid sequence of an encoded protein. And if it does, what extent does this effect have? There'll be a link to this assignment in the video description below. The assignment is going to consist of 3 sections. The first section involves the transcription and the translation of a wild-type DNA sequence. It's represented as a double-stranded DNA flanked by a promoter and a terminator. You're asked to start by transcribing the DNA template into a messenger RNA sequence. The idea is for you to mimic the RNA polymerase, which moves along the double-stranded chromosomal DNA looking for a promoter sequence. This will determine which of the 2 strands of DNA becomes the DNA template. In this assignment, the bottom DNA strand is the DNA template, as shown here in red, and it's in the 3' to 5' direction. The RNA polymerase will briefly separate the double-stranded DNA sequences while it moves along the DNA template in a 3' to 5' direction. The RNA nucleotides are brought in and chemically bonded together during this process using the information based on the DNA template as shown here. Comparing the DNA template to the messenger RNA sequence, you can see that the RNA polymerase will bring in a uracil when there's an adenine on the DNA sequence. Likewise, it'll insert a guanine when there's a cytosine on the DNA, a cytosine when there's a guanine, and an adenine where there's a thymine. As you can see, the order of the DNA nucleotide sequence present in the DNA template will determine the order in which the messenger RNA nucleotides are added. Also note that the DNA template and the messenger RNA are anti-parallel to each other. Transcription ends when the RNA polymerase reaches the terminator sequence, thus releasing the messenger RNA. You're essentially going to do the same when you write the messenger RNA sequence in your assignment. Begin by filling in the first and the last boxes with either a 5' or a 3' to indicate the direction in which the messenger RNA is being synthesized. Next, fill in the remaining boxes with complementary RNA nucleotides using the rule shown here in the top right corner. Group the messenger RNA codons beginning with AUG by either circling them or underlining them. This will help you translate the messenger RNA sequence into an amino acid sequence in the following steps. Recall that eukaryotes form pre-messenger RNA intermediates and only modify them to form mature messenger RNAs, whereas prokaryotes form usable messenger RNAs without the pre-messenger RNA step. This assignment is going to ignore the modifications and focus mainly on the translating of mature messenger RNAs into a protein. During translation, ribosomes will bind to the messenger RNA at its 5' end and move towards the 3' end. In doing so, it's looking for the first AUG nucleotide sequence, also known as the start codon. When the start codon is in the right position in the ribosomal complex, a transfer RNA with its corresponding anticodon will bring with it a methionine amino acid into the ribosomal complex at the receiving position, also known as the peptide site. The genetic code pairs AUG with methionine for this reason. It's telling you that when the ribosome sees AUG, the incoming tRNA with a methionine amino acid will be brought into the complex at the right place. From this point on, the messenger RNA nucleotides to the right of AUG are also going to be read as codons. They will be grouped into 3s. Respective transfer RNAs will compare their anticodons with that of the messenger RNA codons, and it will determine which tRNAs will bring in their specific amino acids into the ribosomal complex. In this example, the transfer RNA that complements the codon CGA will always bring in an alanine to the complex. The amino acids are covalently bonded to each other by forming peptide bonds. They will release the uncharged transfer RNAs, allowing them to leave the ribosomal complex. This process continues as the ribosomes move towards the messenger RNA's 3' end until a stop codon is reached. At this point, the ribosome subunits will separate. They release the messenger RNA and the newly formed amino acid sequence. Like a ribosome, you're going to start at the 5' end of the messenger RNA and look for the first AUG sequence. This will be your first codon. You'll underline it or circle it and group the nucleotides to the right of AUG in similar groups of 3s. You'll use the genetic code to translate the codons into their respective amino acids. You'll write the amino acid abbreviations in the boxes provided below, and don't worry if there's more boxes than you actually need. When you reach the stop codon, you can either fill in the box with the word stop or just leave it blank. Now that you completed the transcription and translation of the wild-type DNA sequence, you're going to repeat this with 6 mutant DNA sequences. Each of these will be followed by 3 questions. And I provide this information in the assignment to help you answer these questions. All 6 mutant DNAs will have one of the following different types of mutations listed here. Here's an example of how you're going to complete the questions. Start by comparing the wild-type DNA to one of the mutant DNAs. What type of mutation does this represent? In this scenario, the mutant DNA has one extra nucleotide that was added. So that, by definition, is an insertion deletion. Next, compare the mutant amino acid sequence to that of the wild type's amino acid sequence and ask the question, how did that mutation change the amino acid sequence? In comparing these amino acid sequences, notice that the mutant has more amino acids in their sequence. So you're going to circle amino acids were added to the wild-type sequence, changing the total number of amino acids. But you can also notice something else, that the amino acids after the site of mutation are different, but whereas those before, that meaning to the left of the mutation, are the same. So there's another answer choice that you should circle, and that is that only the amino acids to the right of the mutation has changed. So the directions say circle all that apply. This is an example where there's more than one correct answer. The last question is not in the form of a question, but it says to classify the mutations. So you're going to circle one of the following: silent, missense, nonsense, frameshift, or no frameshift. The fact that one nucleotide was added to the mutant DNA that changed all the amino acids to the right of that site, so that leads to the codons being regrouped differently than that of the wild type, forming a frameshift. So this is classified as a frameshift mutation. You're going to do this for all 6 mutations. That will complete the first part. Now the second part consists of 6 follow-up questions. I'm showing them here. I'm not going to go through them, but they are part of your assignment. After you complete these questions, you'll go on to the third and final part of the assignment. At this point, you should be familiar with how a DNA mutation can change the genetic information needed to generate a messenger RNA, which ultimately is used to create a protein amino acid sequence. So in part 3, you're going to put your knowledge to the test by creating 5 of your own mutant DNAs. We're going to prompt you on which type of mutations we want you to create. So you're going to start by first transcribing and translating the given wild-type DNA strand. You'll then mutate the wild-type DNA sequence following the directions. For an example, you might be asked to create a silent mutation based on that wild-type DNA. So what you can do is first fill in the boxes with the wild-type DNA and then form that silence mutation. You could change one or more of the nucleotides to form that silence mutation. There's many versions. You get to choose which one you want to present. After filling in the mutated DNA sequence, you're going to transcribe that into its complementary messenger RNA strand, and finally translate that into its amino acid sequence. You'll repeat this process by mutating the wild-type DNA strand to create a missense mutation, a nonsense mutation, a deletional mutation that leads to a frame shift, and finally a deletional mutation that does not result in a frame shift. And that will complete the assignment. So as you can see that the assignment is there to build on the understanding of how the flow of information moves from the DNA template to that of the messenger RNA sequence, and how this may or may not affect the protein's amino acid sequence. It should also give you a good understanding of the effects that various DNA mutations may or may not have. So recall, not all mutations will be seen at the amino acid sequence. And with that, that completes the assignment.